A vaccine is a biological preparation that is used to prevent disease by building an immunity response to prevent the disease. In this discussion, we will be focusing on the West Nile vaccination of equine. A vaccine can hold proteins of a pathogen, weakened forms of a pathogen, or killed forms of a pathogen. For example, to the left is a killed flavivirus chimer vector compared to the right, which is a live canary pox vector. West Nile vaccinations are given intramuscularly to equines, which allows the passage for the vaccination that is given to enter the body and to begin stimulation of its adaptive immune response. When the virus enters the body, it will first be presented on different antigen-presenting cells such as macrophages, dendritic cells, and B lymphocytes, which are cells that are located in your body. These antigen-presenting cells will detect that it is a pathogen and it will be eaten by the cell. When the vector is digested, it will create antigens that can be presented on the antigen-presenting cell surface that are recognized by T and B cells, which are the two major groups of your cell-mediated immunity. This initiates a signaling cascade so the T and B cells both replicate. The B cells will then create antibodies, which are proteins that bind the antigens they recognize on cell surfaces in your body. In this case, it will be signaling death of the infected cell. The T cells secrete signals to different cells in your body, which direct them to target certain cells and perform certain tasks like a supervisor at work. B and T cells both create memory cells that remember antigens of the vaccination, so if your body is infected with the pathogen again, it secretes antigens that notify the body to target and kill the pathogen.